neurological disorders, particularly the ones which are which have no treatment available. Amyotrophic lateral sclerosis, diseases caused by dysfunction in RNA binding proteins. And our goal was recently expanded by our strong collaborations with pediatric neurologists when we started doing identification of rare neurogenetic disorders and their causative agents. So I am going to talk about a successful story today that we started in 2017-18, and then I would like to talk about our efforts, how we approach towards identifying the molecular mechanisms behind this particular syndrome. And at the end, in few minutes, if I have the chance, I would like to discuss about our efforts to identify genetic modifiers and therapeutically relevant pathways. So I am not going to spend too much time on the introduction part, but rare diseases, as Professor Pande said, they are not as rare as we think. There are many of them, 70 million that he was mentioning. But if you talk about globally, there are 400 million people who are affected by this condition. And this is such a devastating number, and there are no treatments available for majority of these conditions. So my journey with rare genetic disorders started when a colleague who is a pediatric neurologist at our University of Pittsburgh, she came and she knocked my door unexpectedly, and she started talking about a patient she was following up. This girl, in the front, who started showing some kind of neurodevelopmental issues, and this patient had abnormal gait, motor dysfunction, was highly ataxic, and has some kind of loss of muscle tone. So every test that was available in the textbook, we tried to do it, and this patient came out to be negative. Negative for mitochondrial dysfunction, metabolic disorder, ataxia panel, triplet repeat, anything that you can think of. So only thing we had was a patient with symptom. We labeled it undiagnosed neurological disease and no diagnosis. So that allowed us to do genome sequencing, and using that approach, we identified a variant of unknown significance, and this was leucine to proline change in one of the genes which is called GEMIN5. And this is what was title of my talk, identifying hidden gems. Because at that time, just to protect the identity of this project, we decided to name it GEM, okay, hidden gem. So parents were heterozygous, and patient was homozygous, suggesting an autosomal recessive inheritance pattern. And furthermore, we found that this patient also have evidence of cerebellar atrophy. So when we started this project, there was only one patient in hand, and no report was published, at least public domain, to support any link between GEMIN5 and any human being. So we found it very interesting, and we started contacting people around the world to see if anybody has ever seen anything similar to this. So nothing came back for another 6 months to 8 months. But one day I got an email from Germany, and this was patient family number 4, where this pediatric neurologist, he had 2 patients, and both patients died within 2 months of age, and they had a variant which was very close to where we identified, with similar clinical picture, with similar inheritance pattern. So we had 3 patients, and I was highly motivated to start doing functional analysis of this variant. Next month we found another patient in France, and after that I stopped counting, and we had significant number of patients at that time that warranted more study. And this is for students, and if you align these variants across different species, these variants are conserved, suggesting that nature, these variants must be doing something very important. That's why the nature decided to keep them even throughout thousands of years of evolution process. So we found there is evidence of cerebral atrophy, and most importantly, these variants were absent in any publicly and privately available databases. So now we decided to publish this report in Nature Communications. We went ahead and we did some functional analysis, and so that clinicians can know about this syndrome, and they can start using GEMIN5 as a target gene in their undiagnosed neurological disorder patients. So now these are the symptoms. And these variants that we identified, they were all over the gene, making it very difficult to do any genotype-phenotype correlation. So we initially had missense mutations that were inherited as a autosomal recessive manner, but over time we identified patients with compound heterozygous mutations. And these patients had different degree of disease, not very homogeneous, but about 80 to 85% of patients, they had infantile onset of disease. And in 42 patients that I am talking about only 15% has age of onset after a year. Very rarely we found any patient with adult onset or who go up to adult. So when I started from Pittsburgh, we have more than 95 patients in 25 different countries around the world, and this number is increasing. Every month I get another new patient. So everybody had one patient and they thought this was not important. And when we published, I think now we are getting more and more patients. So what is Gemin5? So this is a multifunctional gene that has been shown to regulate RNA metabolism as well as translational machinery. So it has been well studied protein at physiological level, where a lot of groups they did analyze the role of Gemin5 in splicing machinery. So as we know, this splicing machinery, spliceosomal complex, is made up of two components. One is RNA, one is protein. That's why people call it, it's a small nuclear ribonuclear protein complex, snRNP complex. So when nascent RNA is generated by RNA polymerase II, this is rapidly capped methylation. There is a methylation cap at the end part of this, and that is a signal that this needs to be transported outside the nucleus. And once it is transported outside the nucleus, Gemin5 is a protein that recognizes this, and this whole recognition process is further supported by other proteins which are part of SMN protein complex, and there are multiple gem body proteins, and there are multiple RNAs which form this complex, and once this whole complex is formed, it becomes mature and it is transported inside the nucleus by another group of protein which is called importing proteins. So over there it sits down in the Cajal bodies before performing its lysosomal functions. So this protein is mainly present in cytoplasm, but there is some evidence of this protein in the nucleus. And also this protein has been linked with regulating different aspects of translation process. So when we started this project, this is the complex that we are talking about. One of the proteins that was well studied in this complex is called the survival motor neuron protein, SMN protein. And loss of function mutations have been reported in this gene, and that has been linked with a disease called SMA. And there are two versions of SMA, SMN protein, which is SMN1 and SMN2, and there is only one nucleotide change in SMN1 versus SMN2. And that one nucleotide change makes SMN2 dysfunctional. And nature designed this problem, but nature gave us a hint to fix this problem for SMA patients. So recently, there are groups who designed antisense oligonucleotides that can fix this one nucleotide change in SMN2, and fixing that would increase the levels of SMN, and patients who would die within a year of age, they are going to survive, and they indeed survive. So this is a miraculous treatment that is available for SMA patients. But in our gene, Gemin5 is a part of this complex, but most of our patients, they presented with a different type of syndrome, where we had motor dysfunction and cerebral atrophy. So then we decided to do further analysis of these mutations and how these mutations are causing disease symptoms in our patients. So we took blood samples from patients who were seen at our clinic, and we isolated PBMCs. We reprogrammed them to become induced pluripotent stem cells. We took another patient who died within a few months, and we used CRISPR-Cas9 to induce those variants in a control cell line. and these cells, they have gone through stringent quality control machine assays. And once we were satisfied, we started asking simple questions. What is the impact of these variants on the subcellular distribution of Gemini? So using that strategy, we found that you can see this is control iPSC neurons, and there is Gemini, which is both cytoplasmic and the nucleus. Whereas if you have disease-causing variants, there is there's a significant decrease in the cytoplasmic expression of Gemini protein. So that is further supported by our immunoblot analysis, where we took these cells, we grinded them, we stained them for different proteins, including Gemini, and this is unaffected individual and from the same family. This is patient, and you can see Gemini protein is barely detectable. Not only Gemini protein, but the whole complex starts going down, meaning that when Gemini protein levels are reduced, other proteins, they also start going down. So this is one mutation. This is the second variant that we have. Protein is barely detectable in these iPSC neurons, and also the other complex members of SNRMP, they go down. So then, next thing that we wanted to ask: why to bother? Why to make this iPSCs? Can we do a rapid test where we can take just PMCs and detect this kind of changes? So indeed, this is in process, but n is equal to 1, like a rare disease, we have evidence that we can detect these changes. So then, next question that we wanted to ask, is it the mutation which is causing reduction in the complex proteins, or can we mimic this situation by genetically knocking down endogenous Gemini in any control cell line? So we took advantage of hex cells and we depleted endogenous Gemini. And doing so, we found that other components of this whole complex, they started going down. So it appears to us like these variants are causing the dysfunction by a potential loss of function mechanism, where Gemini protein is not available and it's not there to perform its function. So what are the downstream consequences? In order to do that, we decided to do in vitro assembly assay, where we labeled one of the components of this complex, SNRP complex, and then we took cytoplasmic lysates from iPSC neurons from control as well as from patients, and we incubated them to see how reliably this complex is formed. So using that approach, we found that there is indeed, this is control, these are patients' cells, and there is indeed a reduction in the complex formation. And not only that, when we did biochemical interaction studies, we found that these mutants, they have limited ability, or their ability to bind to other components is significantly reduced, which you can appreciate over here in the last line. So then, now we have a new syndrome, which is caused by dysfunction in Gemini gene. And while we were studying Gemini, when we published it, other groups started contacting us with newer rare genetic disorders, with hope that we can help them in identifying the mechanism of these variants. So we got another syndrome, which is called Gemini-4 syndrome. And all of these patients with Gemini-4 variants, they had symptoms with everything that we have seen in Gemini-5, but also they had skeletal abnormalities, suggesting that there is something else is going on, which is now part of our ongoing study to understand the molecular mechanisms. But so far, what I talked with you is in vitro data or patient data. What is the relevance of these findings to animal model system or in vivo model system? Is there any way we can identify some pathways, or we can identify some targets that can be potentially modulated by using any approach, and that can be used to develop a therapeutic intervention? So in order to do that, we started using Drosophila as a model system, which is one of our expertise, and we use Drosophila as a complementary model system to iPSC neurons, mouse model, as well as a neuropathologist who is present at our university. So Drosophila model has many advantages and many fundamental biological pathways were discovered in Drosophila, including sleep cycle. A few years ago, somebody was given a Nobel Prize who identified this circadian rhythm in Drosophila. So you can see if you take males and females, put them together, they go through different developmental stages. And this developmental paradigm is fully uncovered, meaning that we know every minute of their developmental paradigm. So this can be potentially used as a model organism system for studying neurodevelopmental disorders. Gemini 5 is conserved in Drosophila and it is called rigor mortis. So we took loss of function alleles of rigor mortis and we did the same experiment that I showed you before, where this is control and these animals, they go through different developmental stages. But if you reduce the levels of endogenous rigor mortis, which is human Gemini 5, there are two observations that we made. Number one, these animals, they have developmental arrest. They take too long from going from one stage to another stage. Second is they die in late pupal stage, very few papers we got. So this was an interesting observation as a geneticist because we can use it as a readout for identifying another gene that can bypass loss of function in Gemini 5. So not only developmental defect, we have evidence that these animals have motor dysfunction and they have reduced lifespan. So in order to identify genetic modifiers, we use the power of our Drosophila model system and we started modulating genes that are part of this SNRP complex, which you can see in the middle of this slide. So since the complex is conserved, we started taking alleles that can overexpress or that can deplete endogenous levels of these targets. So after doing a series of genetic screen experiments, we found this. So this is control, this is rigor mortis RNAi, where animals die in late pupal stage. When you overexpress SMA in SMN protein in these animals, there is a black and white gene, meaning that these animals, they start surviving. So that was a big breakthrough for us. And so question that came up, whether it is only happening in Drosophila or it is true in human patients. So we quickly decided to go in human related cells or human patients. And just to give you a little bit more background about what we were trying to do is, there is therapies available, or there are drugs available that can upregulate the levels of SMN protein. And this has been used in human patients and have been somewhat effective. So these drugs can be antisense oligonucleotides that can fix one nucleotide in exon 7 of 7q, or there are some small molecules available. So we asked this question: what is the relationship between Gemini 5 and SMN overexpression? So just forget about mutation. So we took control cells and we started upregulating SMN genetically. And you can see when you upregulate SMN in control cells, there is increase in Gemini 5 levels. So when you upregulate SMN, Gemini 5 goes down. When you have mutations in Gemini 5 goes down, SMN also starts going down. So it appears like these two proteins have some kind of equilibrium or a relationship that each proteins are tightly regulated. Then we got antisense oligonucleotides which have been used in human patients, and we treated these cells with this control ASOs, and then we used different doses of ASOs, and you can see there is a dose-dependent increase in Gemini 5 level in these cells. So now next thing that we wanted to do, I was presenting actually this data in a meeting in Vienna, and there